Hello, can you hear me? Good morning, good afternoon. I think we can start. It's 3:05, so we had this time for the short late, but time is running, so I think that this is the high time to start.
Before the welcome, one technical note for the English program second year medicine students. If you haven't yet signed the presence list, I have the list after the lecture, all right? Yeah, thank you.
Okay. It is a great honor to welcome you in the 7th. I've counted a few minutes before the meeting, so it's 7th common meeting with the science by now, so far. I am really proud of you. I am really extremely happy to see again that all the chairs are full. So we have many students over here, and again I would like to emphasize that it's not because of the extra points, but you are students of the Pomeranian Medical University in Szczecin. So I know that you are so into the lectures and you are so interested in the different areas of the science to listen to the new lectures. This is additionally that I would like to emphasize that this is the great opportunity to have this new cooperation, science cooperation. You can talk with the great person who we've got today. She's our guest. And I'm sure that this lecture is going to be also amazing as she is. Jennifer Kirwan from Berlin Institute of Health. Before Jennifer will start her lecture, I would like to invite here Professor Vice Dean Karolina Skonieczna-Żydewska, and she will tell us some few words about our guest.
Yes, hello, my name is Karolina Skonieczna-Żydewska. I am very pleased that we have also here health sciences faculty members, so warm welcome to dietitians, cosmetologists, future cosmetologists, who else, biotechnologists and others. So it is my great pleasure to introduce to you Jennifer Kirwan, who has been a very close collaborator of mine for 3 years. We met during COVID when we first met online and were thinking about how and what we should apply for, how much money we should apply for in European Union. So eventually we delivered a project to the European Union during which we are now elaborating on metabolomics. Jennifer is working in Polish cohort of medical workers and others who serves their life to human health throughout their life. So by training, Jennifer is a veterinary science. She originally comes from London. You will have a chance to hear her brilliant British accent. But a couple of years ago, as far as I remember, she moved to Berlin, where she works at Charité, Berlin Institute of Health, and also medical Max Delbrück Center of Molecular Medicine. So she is the very well-known, tremendous specialist in metabolomics, which is one of the very well-known omic technique that is leading the science today. So please give a warm welcome to Jennifer, my friend.
Thank you very much, Carolina. And thank you to the organizers for inviting me today. It's a real honor. I'm looking at the hall full of people. I don't think I've ever had such a full hall to speak to. I'm wondering, what time is Beyoncé coming? I understand that you've had a year of fantastic lectures from well-known celebrities of the scientific world. And then you've got me. But I hope that even if I can't give you the best lecture you've ever heard, you'll at least go away with a good understanding of metabolomics and what it can do for you. So let's discuss what metabolomics can do for you. Can you all stand up? Okay. Now do this. Okay. Turn around. And then on the count of 3, I want you to say yes. 1, 2, 3, yes! You can do it much louder than that. 1, 2, 3, yes! You've just changed your metabolomes. Sit down, please. So this does 2 things. One is it's a very good description of how quickly and how efficiently our bodies respond to changes in circumstances by changing all of our chemical reactions within them. And secondly, it gives me the chance to look at you all looking very embarrassed at the same time. Thanks very much, everyone. Now you know how I feel. Okay, so let's start with metabolomics. I work at the Berlin Institute of Health, and we're mainly interested in precision medicine and medically based metabolomics research. The average drug pipeline for new drugs takes between 15 and 20 years to get from research stage to in the clinic. 15 to 20 years. Some of our patients don't have 15 to 20 years to wait. And this is one of the things that we are trying to do with metabolomics, is try and more efficiently diagnose diseases, predict outcomes, and better target which drugs are going to work in which people. And this is what precision medicine is as a goal. We're working on some really nice projects, such as Immediate with Carolina. Carolina, by the way, super smart person, very well worth getting to know, much more intelligent than I am. The secret of being successful in research is not to be intelligent yourself, but to find the intelligent people to work with. So we've got a great team. Frederica's working on multiple sclerosis. Daniela's started recently with immediate and inflammation. Ayla is doing some nice work on mitochondrial metabolomics. And we're going a lot into new technologies. So we're working in trying to reduce volumes, so getting towards single-cell metabolomics or organelle metabolomics. But we're also moving into metabolomics imaging. So DESI stands for desorption electrospray imaging, where we get these pretty pictures. But quite by accident, I got quite into quality management in metabolomics. And that's because metabolomics, as you've just seen, changes easily. You stand up, you sit down, everything changes. And so we've got people working on bioinformatic tools to help us with filtering and cleaning data. But we've also got chemists working on things like how light-sensitive metabolites are. This is our newest project to see whether we really need to be worried about the lighting conditions in our lab. And can we better identify lipids and more accurately identify lipids? So I've been talking a lot about metabolomics. Let's talk about what metabolomics is. How many of you have heard of metabolomics? Nobody. Okay. How many of you have heard of genomics? A few of you. Okay. So metabolomics is the measurement which includes identification, quantification, and global measurement of small molecules in a cell or a biological system. And by small molecules, we're typically talking about less than 1,000 Daltons in size. We get up to about 2,000 Daltons if we're doing lipidomics, which is the equivalent study in lipids. When we're talking about metabolomics, we're talking about molecules that you're probably very familiar with. Glucose, cholesterol, amino acids, fatty acids, nucleotides, carboxylic acids. So the concept of measuring metabolites is not new. What makes metabolomics a relatively new technology is that rather than doing it on the scale that you're currently doing it in the clinic. So I believe we've got some medical students here. Hands up if you're a medical student, yeah? Okay, so if you send your patients for a blood test, how many metabolites are you normally ticking the box to measure? 9, 10, 11? Let's see, cholesterol, urea, glucose. Shout out a few others. Insulin, well, insulin's a protein, but good guess. Proteomics is a totally other field. But we typically keep a fairly small number, whereas what we're trying to do is look on a much wider scale of typically between 1,000 to 3,000 features, sometimes up to 10,000 features. And you'll quickly see that we have a really wide physicochemical space here, because there's a massive difference between a lipid, which is hydrophilic and quite a large molecule, compared to a small sugar, which is very hydrophilic and much smaller in size. And this is one of the major challenges of metabolomics. How can we find unbiased ways of measuring things altogether? That's a whole nother lecture, but keep that in mind as you go forward with your research. However, metabolomics is really useful because I hope by the end of a couple of slides, I'm going to have proven to you that it's one of our best ways of having an objective measurement of the phenotype of a cell or an organism. So typically, if you have 2 cells in test tubes with genetic differences, we can see this with the metabolites that they're producing. So some of you have heard of genomics. This is the study of the genome, and we're looking at genes. Genes go into RNA. That's called transcriptomics. And the RNA makes proteins, and that gives us proteomics. And at the end of this long chain, we end up with enzymes, and it's these enzymes that are busy making all of these metabolites, which gives us metabolomics. And we'll see 2 things here. First of all, that the time scale of change is quite different between genomics and metabolomics. So typically, to change genomic expression, we're talking about hours to days. It gets a bit quicker when we're talking about translation with RNA. We're now talking about minutes to hours. But by the time we get down to metabolomics, we're often talking about fractions of seconds. And this is why simple things when sampling, like has your person eaten before sampling? Have they rushed to their appointment? Is it morning or afternoon? This is all detectable in the metabolome, as is some of the wider exposome. So the metabolome is largely determined by genes and diet, but is also heavily influenced by your lifestyle, how social you are, for instance, by your environment. And we can talk about local environment, Do you live in a city or a rural area? Do you live near a coal-fired power plant? But it's also very local environment, such as, are you spraying perfume on yourself? That then gets absorbed, and we'll measure it in your metabolome. And I've highlighted microbiome because this is a special interest of ours and of Carolina's, so it's a big issue in the metabolomics world at the moment. So we're particularly interested in metabolomics and inflammation. How many of you have experienced inflammatory disease? How many of you have ever had a spot on your face? Wow, some of you have really good skin. This is the typical model of inflammatory disease. You have an initial trigger. You get a collection of immune cells rushing to the site, and you have what in English is known as the cardinal signs of inflammation: redness, swelling, heat, pain, and loss of function. And what we can see is that between the initial trigger and immune cells flooding to the site, something happens. Magic. And between the immune cells causing inflammatory disease and then the inflammatory disease receding, something happens. Magic. That's for intelligent people to study. I'm not particularly intelligent, so I look at the more simple things, which is what controls the process in between. And in particular, if we look at metabolomics and our traditional model of inflammatory disease, we start to see that it breaks down a little. So it works very well for local, visible inflammatory disease. I go skiing, I twisted my knee, it hurts a lot, it swells up, it's very clear what the trigger is, and it's very clear what the outcome is. But we are also increasingly finding that we see that there's inflammatory processes involved in things like heart disease, Alzheimer's, potentially some psychiatric diseases. So single organs seem to be suffering from inflammatory disease, but the triggers are not always known. And in some cases, we're finding that where the triggers are known, in some people they're causing heart disease, and in other people they're causing kidney disease. And we also have this issue of multi-organ and systemic inflammatory disease. What causes an inflammatory trigger to go from a single organ to a multi-organ state? So inflammatory disease in itself is not my speciality, but I am interested in how metabolomics contributes to it. And we're also interested in what causes an acute inflammatory process to end up as a long-term chronic disease. So we're particularly interested in the microbiome in this process and in how different organs in the body are cross-modulating this process. So if you have heart disease, you're much more likely to have depression. And this was always thought just to be an effect of illness, but we're now increasingly realizing that this actually seems to be linked to the same inflammatory processes. And so we're particularly studying this gut-heart-brain health link. And we're doing this through some large cohort studies. So we have the BLOVE study in Berlin. This is studying long-term vascular events. And this is particularly looking at if people have heart disease, why are they more likely to get kidney disease or strokes than normal healthy populations. We're looking through the immediate project. This is an EU-funded project at why people end up with inflammatory disease and which inflammatory diseases, which markers of inflammatory disease are the same across different cohorts and which are different depending on which diseases they have. And we also have the national German cohort, or Nauką, which is looking at healthy people and tracking them as they get disease. These are all large cohort studies of between 2,000 and 10,000 participants. It's a challenge in itself just to deal with this number of samples. And if any of you are interested in the biotechnology of that, do feel free to come and chat to me afterwards. So we're particularly interested in tryptophan in our lab, and this started way back in my Birmingham days. This is Niamh Dennehan. She was my first PhD student, and she was a student of Deirdre Murray at UCC in Ireland, and they were looking at hypoxic ischemic encephalopathy. This is a disease that you get when you suffer a traumatic injury at birth. This is when the baby suffers lack of oxygen. Did I just say a traumatic injury? Sorry, a traumatic birth. When you have a lack of oxygen to the brain and you end up brain damaged. Highly distressing to both the parents and the child. There is a treatment, but it's not 100% effective. It has a lot of side effects. And Niamh and Deirdre approached me and said, can we use metabolomics to work out who needs to be treated? Now, there's an intermediate condition called perinatal asphyxia, and this is characterized by the baby going blue at the time of birth, but it doesn't progress to severe brain damage. So they had a great cohort recruitment drive at UCC, and this is quite a rare disease in Ireland, but they ended up with 41 perinatal asphyxia cases and 30 HIE cases. They were tested to a gold standard, and they had 41 match controls based on 5 different matching criteria. We were using direct infusion mass spectrometry, which is quick but dirty, and we were able to collapse the results down into looking at different metabolic pathways. And what you'll see from this list is that although Krebs cycle is up there, energy production, lack of oxygen, not a surprise, so is the pyrimidine pathway, and so is the tryptophan metabolism. And what particularly excited us here is that these seem to be incrementally different between perinatal asphyxia and HIE. So we investigated this further. Now, this is the tryptophan pathway, highly complex, and this is only a part of it. Most of you will have heard of some of the tryptophan metabolites, such as serotonin, melatonin, 5-HT. They're very, very important in the brain. But as you'll see as we go through, they have other roles. What we got very excited about here is how many of them were changing, and that at least some of them seem to have differential changes between HIE and perinatal asphyxia. Now, I left Berlin, but they had enough money for a follow-up study, and this time it was a cross-clinical study between UCC and Karolinska. And out of the original 33 metabolites for targeted validation, they were able to validate 15 of them across the 2 groups. This is called a spring-loaded plot. I love these. For those of you who are less familiar with these, this basically shows what's correlated with what. And what we see here is tryptophan is very nicely on the left-hand side of the plot there in red, and it links very nicely to the APGAR scores. Now, APGAR scores are the scores that the doctor gives the baby within a few minutes after birth to say, is the baby breathing normally? Is the baby moving normally? It's a very quick check of how healthy the baby is. And we see that they're also highly correlated with outcome. This work was done by Dara. He was a master's student of Deirdre's, and he was able to show that using a type of modeling called random forest, by using both the clinical data and the metabolites, he was able to get a much higher sensitivity and specificity than just using the current gold standard clinical data. In addition, what we saw was that kynurenine and tryptophan, which are two metabolites from the tryptophan pathway, both seem to be important in the metabolite model of the data. And there was some evidence that not only could it differentiate between HIE and perinatal asphyxia, but also between different grades of HIE, but we didn't really have enough sample size to confirm that. What was exciting for me was this issue with kynurenine and tryptophan, because what I haven't told you up until this point is that this was now the third neurological study that I had been involved in where tryptophan and its metabolites were coming up as important in diagnosis. So let's look at tryptophan in more detail. Tryptophan is a really unusual amino acid. It's one of the rarest amino acids, and where it is found in the body, it tends to have a structural role. It's metabolized by our body either mainly in the liver or in the gut, but it also ends up a lot of it in the brain. And it has two major metabolic routes. Either the indole ring is broken, this is this second ring there that you see, and it ends up as a metabolite called kynurenine. Or the indole ring is maintained, and it ends up as a lot of neuroactive compounds. And we're back to serotonin, melatonin, 5-HT. Now, we can get a little bit more complex when we look at what the gut microbiome is doing. And the gut microbiome metabolizes about 5 to 10% of our dietary tryptophan. Doesn't sound much, does it? However, what we find is that it's metabolizing this tryptophan into lots of very different compounds, and these compounds are very bioactive, and these compounds are doing everything from being anti-inflammatory to activating the aryl hydrocarbon receptor. The aryl hydrocarbon receptor is this weird figure here. I don't know whether... No, okay, the laser pointer. Okay. The aryl hydrocarbon receptor is this weird colored figure here. Take note of this, because this is very important, or it is if you're a researcher. The aryl hydrocarbon receptor has a definitive role in deciding whether your immune system is going to be primed for attack or whether it's basically just going to be on guard duty. Making it even more interesting is when we look at what's doing what. Different species in the gut seem to be metabolizing tryptophan to different compounds. And this means that the composition of our gut microbiome really makes a difference in what's happening in our immune system in the local part of the gut, and as you'll see later, in the general immune system as well. So I'd moved to Birmingham by this stage, and I'd moved to Berlin by this stage, and our group then spent a lot of time and effort in putting together a dedicated tryptophan metabolite assay. How many of you have heard of mass spectrometry? Ah, good, we've got a few, okay. So this is a mass spectrometry assay. It's fully quantitative, it's validated to EME guidelines for serum and for urine. We've tried a few other interesting matrices, such as gut mucus, and we're increasingly finding more and more interesting results from it. This is Dai Long and Matthias Kuring. They're both former colleagues of mine, and Ulrike Bruning also contributed. She's a current colleague. So once we had our 35 metabolite panel, we needed a pilot study to test it on. So step forward, Dominik Müller and Johannes Holler, and Nicholas Wilk and Felix Behrings from the Charité. And they came to me with a question: why are our children dying of heart disease? They were nephrologists. They were interested in kidney disease, and what they were seeing was the children with kidney disease had a much higher risk of heart disease. This was already known about in adults, and it's the leading cause of death in children with end-stage kidney disease. Now, in adults, it had been shown that the gut microbiome was disturbed, and there was an assumption that this dysbiosis was causing a buildup of toxic metabolites, and this was further poisoning the kidneys, and this was leading to the heart disease. So we ended up with this cohort of around 10 patients who were, or 10 healthy controls, I should say, and then we had a gradation of disease. So CKD, which is chronic kidney disease, we had 12 patients. This is the lowest level. Hemodialysis patients is when your kidneys cannot do anything. And by the time you get to a kidney transplant, in theory, your kidneys should be working near normal, and so they should be closest to the healthy control. So we ran our tryptophan pathway, and what we saw was this clear gradation in results. So if you look at the indole acetic acid, this is the second chart on your left there. You'll see that our healthy controls have a very low level, then it creeps up for the CKD, and the hemodialysis, it gets quite high. And then after kidney transplant, it drops down again. The last one on the end is our pool QC, and this is very important for our quality control, and also to see how much tolerance we have in our measurements. This was something that we saw over and over and over again for our indoles. But I'd like to particularly point out indole 3-propionic acid and indoxyl sulfate. Now, indole 3-propionic acid maintains tight junctions in the gut, and therefore it's really important for maintaining gut permeability, or lack of it. Indoxyl sulfate is known to be a kidney toxin. And what we see is that our indole 3-propionic acid goes down and our indoxyl sulfate goes up. However, when we got discussing with the doctors, we started asking about diet, because we only get tryptophan from our diet. It's an essential amino acid. Oh, they said, the hemodialysis patients can't eat much protein. They're on really low protein diets. So we're not strictly comparing like with like. So in this case, what we did was we normalized everything to our plasma levels of tryptophan. And suddenly what you see is that when you look at it in relation to tryptophan, these children are desperately fighting to keep indole 3-propionic acid. They are converting a much higher percentage of their tryptophan to this molecule to try and keep their guts healthy. Unfortunately, it's a losing battle, because at the same time, something in their gut is producing lots of indoxyl sulfate, further poisoning their kidneys. We also looked at tryptophan metabolites that were directly involved in the immune system. These are aryl hydrocarbon receptor agonists, so these are the things that will either be pro-inflammatory or anti-inflammatory, and all the ones up here are pro-inflammatory, and we can see that they're clearly all raised. And this work was done by our collaborators. They were able to show that if you incubate cells with serum from children with chronic kidney disease or hemodialysed patients, that tumor necrosis factor alpha increases markedly. So TNF alpha is also something that sets the immune system going. Indoxyl sulfate was also found directly to be a potent agonist of the aryl hydrocarbon receptor. This was not something that we'd anticipated. And they also showed that there was a switch from classic to non-classic monocytes. So another group measured short-chain fatty acids. This is important because they're regulating the tryptophan pathway. You get these mainly from your microbiome fermenting fiber in your gut. And they also found these were decreased and TMAO is increased. Does TMAO mean anything to you? Looking at the medics in the room? TMAO is a metabolite of TMA, which we get from red meat in our diet, and it causes atherosclerosis in humans, directly linked to heart disease. So it seemed clear that we had this direct link between immune function, TMAO production, and the heart disease we were seeing in these patients. And when we looked at the microbiome, this work was done by Sophia Forsland, we could see that the more proteobacteria that these children had in their gut, the more it was associated with markers of gut permeability, so bad, zonulin 1 and TNF-alpha. However, if we look at the red negative correlation, we see the more Peteschi bacteria you have, the lower the indoxyl sulfate component. So what we can say is that children with CKD have gut barrier dysfunction and dysbiosis, and this bacterial metabolite imbalance is directly contributing to changes in immune cell populations and the activation states. This is leading to stage-dependent progression of immune function, although this can be reversed after transplantation. And the most exciting bit about this work is that this leads us to avenues of potential support for these patients, mainly through the use of pre- or postbiotics to support their gut microbiomes. I'm going to finish up with a final study. This is COVID-19. COVID-19 meant a lab shut down for us, and the only thing we were allowed to work on was COVID-19. So we were all work bunnies, so we threw everything we had at our COVID-19 project. We did gas chromatography mass spectrometry, we did liquid chromatography mass spectrometry, we did kit-based methods. We measured in total somewhere close to 650 different compounds. Our collaborators were looking at viral load measurements and cytokine measurements and PBMC and microbiome, not only of the gut, but also of the oropharynx. And why were they interested? This was Sophia Forsland's group again. Well, this is because we were looking at hospitalized patients. And our biggest overall question was, could we predict which hospital patients would get better and be discharged and which would deteriorate? And we had up to 5 samples per individual over time. So the analysis was done by Morgan Essex. She's a bioinformatician in Sophia Forsland's group. And the first thing she was able to show were lysophosfatidylcholines were clearly dysregulated and reduced no matter what stage of COVID you had, whether it was severe or mild, and no matter what time point we tested. Back to the tryptophan pathway, and she created these lovely diagrams. Take-home message here is that kynurenine massively increases in severe disease, whereas tryptophan decreases, at least in plasma. It's a bit more complex in urine. And kynurenine is positively correlated to cytokines. So for those of you who are not inflammation experts, cytokines are those messengers to tell the immune system to ramp up. We could look at the microbiome and say that Faecalibacterium was correlated to tryptophan. So the more Faecalibacterium you had, the more tryptophan you had, which was protective. Tryptophan was also protective against the number of cytokines you had. She did a lot of work on confounding factors. And she did a lot of work on looking at microbial metabolism. And we see that secondary bile acids were decreased, as were many of our key contenders, the indoles in the immune system. So what we can say here is that lysophosfatidylcholines, they're pro-inflammatory and immunoregulatory, and they decrease in COVID-19. Faecalibacterium is associated with a decrease in immunoregulatory tryptophan, but if your kynurenine to tryptophan ratio goes up, then you're more likely to get inflammatory cytokine production. And our microbial-derived indoles were reduced in severe infection, which also had immune regulation consequences. And this resulted in what I like to call the circle of despair. We ended up with a microbiome dysbiosis, which causes an immune system disruption, which causes further microbiome dysbiosis, which then leads to further immune disruption. But this is really why I'm showing you this study. The most interesting thing for us at the end was we'd measured at least 7 different types of factors. But in the end, it was the metabolites that explained the best proportion of the illness severity. You remember the bit at the beginning where I said that metabolomics is a great way of measuring phenotype? This is where it comes into the real world, where you as a doctor or a researcher are able to make better treatment decisions based on tests that you have available to you. And we can also see that it was the metabolites, and particularly the tryptophan metabolites, and we're back to kynurenine and tryptophan, that were acting as these direct links between our microbiome and our immune system. So I don't know whether I've got time for the last bit. Do I have 2 minutes? I put this in specially for Carolina, because we've had a lot of discussion on stool. So most of our stool comes in bottles like this. If we're really lucky, we get a bit more like this. And if we're really unlucky, then we get a swab. I don't know about you, but my stool doesn't look like this. It looks like this. And in fact, because I really like the Berlin specialty lentil soup, more commonly it looks like this. Now, instinctively, we know that stool is not a homogeneous matrix. And if this is not instinctive for you to know, think about the last time you ate sweet corn. Now, we can start with this instinct, but we also have studies, and this one by Barger et al. Was published in 2012, showing clearly that at least for certain steroids, you get pockets of steroids in the stool. And so I became a bit obsessed with how to collect a representative stool sample. And I hope by the end of this talk, so will you. Because ask yourself, if your stool looks like lentil soup, how is giving a researcher a swab going to be representative? So we built a stool crusher. This is a very simple device, which is basically a heavy weight with a lever where you can put a frozen stool in a heavy-duty plastic bag, crush the life out of it, and in theory end up with powdered stool at the end, still frozen, which can now be freeze-dried for either metabolomics analysis or genomic analysis. And on that note, I would like to invite you to think about how you can make this better, because it doesn't work as well as we would like it to. Job for the biotechnologists of the future. But most of all, I want you to go away with the idea, if you want to do a stool sample study, please collect whole stool and think about homogenizing it. Okay. I think I've said enough. Immune regulation is complex, but tryptophan plays a really major role. And otherwise, I just need to remind myself and you that most of this work was done by other people, particularly the fantastic members of our group. I'd like to acknowledge them all, particularly the people involved in some of the studies. This would be Ayla Fritscher, Frederica Gutmann, Daniel Amitahu and Ulrike Bruning, and of course our funders. Thank you very much, and the floor is open for questions.
Thank you very much for this interesting lecture. I realized how many important information is included in this single stool sample. I mean, it is amazing to discover it and to listen to your lecture and to realize that, actually. I have a question for the public. Ask any questions for our great people.
I would like to comment. There is always a story in the stool. As Jennifer told, we have spent a lot of hours in elaborating on how to collect stool. I have personally delivered stool to the laboratory, picked them from doctors and our patients' homes. So stool is very, very important. When you come to the Department of Biochemical Science, which is located at Broniewskiego 24, and you hear we are doing a poo, it doesn't mean we are going to the toilet to get a poo, but we are doing some lab work with a poo, which is... and in fact, it's not a taboo anymore. When, together with Professor Stachowska, we were going to Woodstock Festival, now Poland Rock Festival, and we were approaching to participants of Woodstock Festival and were asking these people, how does your poo look like? Does it stink? Does it shine? You know, at first they were astonished with the questions, but eventually everybody poo, and if you poo every day, the life is brighter. So thank you. And maybe I have a question, because Jennifer, you told us at the very beginning that frequent sampling can change the metabolome to a large extent. So should we focus in our future research on frequent sampling? Not only in one time point, as we got used to do in our clinical research?
So I think, I mean, I get quite obsessed with experimental design. And I think the key thing is to know exactly what question you're trying to answer when you're designing your experiment. It's great to have multiple time points, but sometimes we just don't have the money or the participants to give us multiple time point samples. So then the most important thing is to think about what your biological question is and how to structure your experimental design and your sample number. I think we have a real problem, particularly with animal studies, of very small sample numbers, which is important for ethical reasons, but we have to ask ourselves whether it's ethically worse to do a study that cannot be statistically, it's not statistically powered, than not to do the study or to do it with a larger number of animals in the first place.
Yeah, I have a colleague who runs a study in cows. We were talking about this a couple of hours ago, and she awarded a consent from the local ethical committee to run a trial in 4 cows. So 2 from the intervention group and 2 from the placebo group. And now, when she finished the study, she has a huge problem to publish the results because of the not enough statistical power of the results. So, yes, thanks.
Are there any questions? Other questions from students?
There's one at the back.
Thank you very much for the fascinating lecture. I have a question about urine, because you talked much about the importance of having representative samples of the stool. What about the urine? Because I think that in some laboratory tests we do use, for example, midstream urine. Are there any rules that apply to research in urine?
That's a super question. So we have two problems with urine. The first is when and how do you sample? So midstream urine is easy, but you risk that it's not representative of the whole 24-hour cycle. The other problem we have is the different dilutions across different urine samples. So ideally, you get a 24-hour urine sample. In practice, almost never happens. So what we try and go for is an overnight fasted urine sample, that at least there's some degree of regulation. That is a bit study dependent. With the dilution, we're moving towards specific gravity as one of the most reliable ways of dealing with the dilution, but there are other ways such as drying urine and analyzing the metabolites that way. The challenge there is that you may not be drying the same amount if it's at different concentrations, and you will change some of the metabolites by the drying process. So that's a lecture in itself, but I'm happy to discuss further if you're working on urine.
Oh yeah, I was wondering, since last year the protein folding problem got kind of solved by AlphaFold 3, if you've worked with AlphaFold or your colleagues, and how has it changed the field of metabolomics?
Another great question. So I think that AlphaFold 3 is massively going to change the field of proteomics and drug discovery. And I think that that will speed up certain links that we can make between metabolites and proteins. In particular, which metabolites may be agonists or antagonists of proteins. I think the field of AI in general will speed up science. We've got small tools that can better predict how to change assays as you switch them between different instruments, for instance. We're using machine learning on a regular basis for looking at differences between different groups. And I think that AI is just going to change our lives. But here is a word of warning for you. AI is great, but sometimes it allows you to switch your brain off. And you need to have a certain level of knowledge to be able to understand when the AI is helpful and when it's decidedly not helpful. And as an example, I asked AI to draw me a picture of the tryptophan pathway, and it drew me a picture of the tryptophan pathway. It had all the metabolites in, but unfortunately not in the correct order. Now, I knew this, so it didn't go forward. But the one benefit that you will have as you're going through your studies is to read lots of papers in their original form. That doesn't mean you can't use AI, but there'll be lots of hidden data there that isn't in the summaries. And if you can amass that data, you'll be amazed at how that can all come together 20 years later when you say, hang on, I remember that Cox was involved in pain, which, real life story. We were taught about Cox in biochemistry. I thought this was completely irrelevant to my life, Cox 1, Cox 2. And then suddenly somebody developed a class of analgesics based on it. So you never know what's going to happen. Read, and read more. That's all I can say.
Thank you.
It's like website. Everybody thinks that it's unuseful, but eventually it is.
another question there.
Okay.
Hi, I have a question because it sounds like we all have very individual gut microbiome, and with all these studies, it looks like the results that we have, the learnings that we have, they're still very kind of individual because we don't know what we have inside. We need to research. So can we think that in the future we will have this targeted but very individual treatments to control either, for example, tryptophan will metabolite into beneficial and not toxic metabolites? So is it a super individual treatment that we're talking about in the future? Because we all very different, we have different background, and we don't really definitely know what we have inside. I'm not sure if my question is clear.
No, I think it's a reasonable question. So we used to talk about personalized medicine, and about, 5-10 years ago, it was decided to change the name to precision medicine. And this because there was a recognition that you can only go down so far before medicine becomes too expensive to do on a very, very individual level. That said, the use of AI and the use of modern technology, you can pay €350 and have your gut microbiome sequenced in a private company. What it means, the results, that's a different question. And I'm not sure that we're sufficiently knowledgeable about the actions of gut microbes that anybody is able to truly say what a healthy gut microbiome looks like and what a particular microbiome will or won't do. We can make correlations, but like anything in the human body, if you change something, it often has unexpected effects. But I think, as we go forward, this is an exciting field, and I think in the next 10, 20 years, real possibility. You get given a prescription of this, and this microbiome.
Because personally, as a medical student, for example, after learning something this year about it, in our subject of clinical nutrition, I personally don't like any generalized advices, like in terms of the nutrition and everything. I think it's very individual, and that's the challenge that I think we're facing in the future.
Come and see me afterwards and I'll tell you a couple more anecdotes based on real life research.
And if possible, I would like to add on, because I have a lecture, right? I don't know if you heard in Moodle on axis, and this is also the area of my research. So we do differ to a large extent on the gut microbiota composition. So as Jennifer said, it's not necessary to analyze the composition of the gut bacteria, but gut bacteria does do the same in every single person. So the most important thing is to analyze the metabolites. So this is the area of Jennifer's work. So by means of the metabolomics, we can analyze what is happening within your gut. We don't need to have the knowledge on what's in the gut. We do not know what the healthy microbiome looks like, but we do know what the healthy microbiome produces. So this is the key issue, I think.
And I think that's a very good point. You may have microbiome X, and Carolina has microbiome Y, and I have microbiome Z, but we may all be producing the same amount just through different microbes.
For instance, I do not like lentil soup, but I'm sure I do not have the same bacteria as you, but we do produce serotonin, I'm pretty sure.
Okay, I think this is time, and we can actually say goodbye to everyone. But if you don't mind, I've just had the idea, and maybe this is the question to ask for the photographer, or I don't know if he's still in this aula, but I would like the common picture, the common photo of us, all of us, and regarding to the beginning of your lecture with the changing the microbiome of us, could you please stand up, everyone, and take your hand up? But I don't know if we have still any photographer. Just a camera, right? Okay, so I'm gonna give you